This lecture, we're going to talk about pharmacokinetics. To understand pharmacokinetics, let's do a thought experiment. So suppose this is a patient, and imagine this patient is a cancer patient, unfortunately. And this patient has pain for which the patient is taking a 30 milligram tablet of morphine. Okay. So this patient is taking, because it's a tablet, obviously the route of administration would be per oral. Okay. Okay, so this tablet, and we are trying to see the course of how the tablet ultimately enters the brain. Okay, so we are trying by this thought experiment to trace what happens to the tablet. Okay, so the tablet reaches the stomach, where it gets where it disintegrates, and then it dissolves. Okay, then it reaches the intestine. Here we're talking primarily about the small intestine. Okay, what are the parts of the small intestine? Right. Duodenum, jejunum, and the terminal ileum. So from the small intestine, it gets absorbed. Absorbed where? Into blood. After it gets absorbed into the blood, it gets transported. Okay. So it gets absorbed into blood, then it enters the systemic circulation, meaning it goes into the liver. In the liver, some part of it gets metabolized, and then it comes back to the blood. Okay, so that is the systemic metabolism. Okay, then it is transported in the blood, and it crosses the blood-brain barrier. In case of women, it can also cross the blood placental barrier. So there are barriers. Everything that is in the blood does not reach certain tissues. Okay, so in the CNS, we have got the blood-brain barrier. The other barriers would be blood placental barrier. And blood testis barrier. Okay. Okay. So in this case, it crosses the blood-brain barrier, and then it crosses it gets into the neurons. And then the pain-killing effect is seen. Okay. So that's how we have traced the morphine tablet. Okay, it ultimately produces an effect where in the CNS. Okay, so in this case, it's a pain-killing effect. So what's important to remember here is in the intestine. Let's imagine this is the intestine. Okay, so it gets dissolved here. So this dissolved morphine is absorbed, and then somewhere here, let's imagine there are blood vessels. So this is a blood vessel, and this is a part of the small intestine. Okay. It crosses into the blood vessel, and through the blood vessel it is transported, and then here again it crosses the blood-brain barrier, which is kind of a membrane here, and then the neurons themselves, say this is a neuron, and these neurons have got their own membrane, and it crosses the neuronal membrane, and then it has got the effect. So what is clear here is that the drug morphine, it has to cross membranes. Okay, in case of morphine, we see that it crosses the intestinal membrane. So let's write it down here. There is the membrane of the blood vessel, and then there is the membrane of the blood-brain barrier. So there is another membrane which makes up the BBB, blood-brain barrier. Then we've got the neuronal membranes. Okay. Then it crosses this membrane, and then it has got an effect. So the important point is that drugs need to cross membranes. Okay, so that's the important point that we need to remember. Okay, needs to cross membranes for their action, to produce an action. Okay. So there are four mechanisms. So there are four mechanisms by which drugs cross. Drugs cross membranes. Okay. Okay. So what are those four mechanisms is what we're going to learn now. Okay. For that we need to draw a diagrammatic representation of the membrane. So this I'm trying to diagrammatically show the Singer-Nicholson model of the cell membrane. Okay. So this model tells us that there are certain proteins. So let's imagine this is a protein here, which cross both layers of the membrane. So this is an integral protein. Okay. So outside this cell, this is the extracellular region. And this is the membrane, and this is the intracellular. region. Okay, so below this part is the intracellular region. Let's draw this membrane like that again, okay, and continue to draw the membrane. Okay. And somewhere here there is kind of a pore, and this pore, through this pore water enters into the cell. So the technical name would be aquaporins. Okay, so this is an aquaporin channel. Okay, so it's a pore through which water can enter, and we can continue to draw the membrane. Okay. So this is a membrane. It's a lipid bilayer. So this is the phosphate groups, and these are the lipid bilayer. There are two layers of lipids, this and this. So this is basically a representation of the Singer-Nicholson model. Of the cell membrane. Okay. So let's say this is a drug. Okay. So drugs, they can be, depending on their molecular weight, they can be classified as small molecules and large molecules. Okay, so small molecules, they've got molecular weight less than 1,000 daltons. Okay, this would be greater than 1,000 daltons. So here, when we say a drug, we mean a small molecule. Okay, though it's less than 1,000 dalton, usually it's less than or equal to 500 daltons. Okay, so most of the small molecular drugs fall within this range. Okay, so this drug can cross the membrane by a process called passive diffusion. Okay, so it can diffuse through the membrane. Okay, so this first mechanism by which the drug can cross the membrane. So let's write it down here. So the first one is by diffusion. Okay. Okay, so passively it can cross the membrane. Okay. So it doesn't require energy. So for diffusion we don't need energy. And it depends on the concentration gradient. It depends on concentration gradient, meaning if the extracellular concentration is greater than intracellular concentration, then we say that there is a positive concentration gradient. Okay, so there should be a positive concentration gradient, and it should be a small molecule. And since it's crossing this membrane, you can imagine that on the top part we've got hydrophilic layer, and the inner part would be hydrophobic. And a small molecule has to cross this hydrophilic layer, enter into a lipophilic layer, and then exit out into again a hydrophilic environment. So there are certain constraints that so this places some constraints on this small molecule. So it should have lipid solubility, and it should have water solubility as well. Okay, so generally it has got this characters, but there are more characters that we will be discussing later on. But for now, we need to remember this. Okay, so by passive diffusion, a small molecule can enter into the cell. It can cross the membrane. The second mechanism is it can use a carrier. Okay, so a carrier is a protein like this. So once it combines with the carrier, the drug combines with the carrier undergoes a change in its conformation, and you see that it kind of flips here and then reaches the drug into the intracellular environment. So the second mechanism, let's write it down here. The second mechanism is by carrier mediated. Okay. So there are different types of carriers. We'll be discussing that again. So one of the most important carrier is SLC. Okay, so this is a solute carrier. Okay, so let's imagine this integral protein. What's an integral protein? Which crosses both the layers. Okay, both the lipid layers. If it crosses, that's called an integral membrane. Okay, which also means there are peripheral proteins. Okay, so there are some proteins which are found only in one layer. Okay, so this would be a peripheral protein. Okay, so a carrier is basically an integral protein. Again, it doesn't need energy. Okay, this carrier kind of is specific. It binds and then it flips and it reaches it. Okay, so the second mechanism is carrier mediated. Okay, so the third one is some drugs can use the normal water channel. Okay, it can use the aquaporin membrane to get into the cell. Okay, it can use the aquaporin membrane and enter into the cell. Okay, so the third mechanism is through aquaporins. Okay, so aquaporin mediated transport into the cell. The fourth mechanism is by pinocytosis. Okay, so pinocytosis is when the membrane. So suppose this is a cell and this is the extracellular part. Let me write that properly. Okay, so this is the extracellular environment. This is the membrane and that is the intracellular environment. Okay, so pinocytosis is when a drug is there, it dissolves in liquid and then the cell membrane kind of invaginates. Okay, the cell membrane invaginates and takes in the drug molecule within the cell. Let's say this is the eukaryotic cell and that's the nucleus. Okay, so that's the nucleus. And we see here that the membrane invaginates and then ultimately it reaches the drug within the cell. Okay, so we've got this cell and here we have the drug and This again is the nucleus. Okay, so the drug which was in the extracellular environment dissolved in extracellular fluid um, um, gets into the cell by this mechanism, pinocytosis. So what happens in pinocytosis? There is invagination of the cell membrane. Okay. So broadly, these are the four mechanisms by which a drug enters into the cell. Okay, so what are the four mechanisms? Diffusion, carrier mediated through aquaporins, and by pinocytosis. Okay, so we'll visit these points with some more details. In diffusion, where the molecule traverses the membrane, we need to remember that non-polar drugs they rapidly diffuse through the membrane. Okay, so that is the reason that chloroform. Okay. Okay. It has got a very rapid onset of action because it easily crosses the membranes. So it can cross the blood-brain barrier and cross, and depress the neurons or neuronal activity. So non-polar drugs can easily cross the membranes. Okay, it's a rule of thumb that we need to remember. Which also means, so this is so another point that we need to remember is that if the drug is ionized, okay, then the likelihood of it traversing the membrane decreases. For example, let's take the example. Let's see if I can paste this drug, okay, this structure, right. So this is the structure of drug. This is, let me write down the name of the drug. It is DTC, okay, curare, okay. So this is a poisonous plant, okay, and the property of this drug is that it's a neuromuscular blocker. Okay, so certain tribes in the Amazon rainforest, they dip their arrows. So if that looks like an arrow, yeah, I hope it does. So what I'm trying to depict here is an arrow. Okay, so they coat the tip of this arrow with curare. Okay, curare. Okay. They have got this plant in the forest and they make an extract of it and then they coat the arrow of the plant. They coat the arrow with the extract, and then they use it to hunt the animal. So suppose, say, there is a deer here. Okay, so imagine this is a deer. Okay, so just imagine that this deer has been wounded by the arrow. So what happens is this curare gets into the blood of the animal. And then it paralyzes the skeletal muscles. Okay. So it paralyzes the skeletal muscles. So basically it's a neuromuscular blocker. Now if it paralyzes the skeletal muscle, the animal will not be able to run far. Okay, it would be paralyzed and it would fall, and then it becomes easy for the tribes to retrieve the animal after wounding it. Okay? But this same animal is later on consumed as food. Okay? But when they consume it as food, they don't find any ill effects. Okay? Meaning their neuromuscular, they don't undergo paralysis. Okay? So the question is why? Okay, when that same animal which has been wounded by curare, so presumably the meat contains curare, and that meat is consumed by the tribe, but when they consume it, they don't suffer the same effects. So why? The reason is that if we look at the structure of D. tuberculare, we see that it is ionized. Okay, so if it's ionized, it is highly ionized, what will happen? It cannot cross the blood-brain, membranes. So meaning it won't be absorbed. Okay, so they may be consuming it, the meat might contain curare, but it won't be absorbed, and therefore if it is not absorbed, it will not reach the blood, and if it does not reach the blood, it won't cause neuromuscular effects. Okay, so that is why they are able to consume. So the point here to remember is that ionized compounds, they usually cannot cross the blood-brain barrier. Okay, so but certain drugs, even if they are ionized, they cross the membrane barrier because they got specific transporters. Okay, but this fortunately, human body does not have a specific transporter for curare. Okay, because of which they can use this plant to aid them in their hunt. Okay, right. But the rule of thumb to remember is if the drug is highly ionized, it's very difficult to cross the membrane barriers. Okay. Okay. Then the other point that we need to remember here is we told that carrier-mediated transport. Okay. So solute carriers. We talked about only one carrier, like solute carrier. We need to remember that this solute carrier is of two types. Okay, it comes in two flavors. The one is organic cation transporters, and if it is cation, then it can be, the other one would be organic anion transporters. Okay. So organic cation transporters, they transport drugs like dopamine, okay, quinine, okay, which is given for malaria, and the organic anion transporters, they are usually involved in the secretion of drugs such as probenecid in the kidney. Okay, so this point will be visited when we study penicillins. Okay, so I think we can learn here. So penicillin is excreted, okay, in the kidney by secretion. So we know there are different mechanisms of excretion in the kidney, that is filtration, secretion, etc. So penicillin is excreted by secretion into the kidney tubules. Okay, so this secretory pathway is blocked by probenecid. Okay, so this is blocked by probenecid. So if you are giving penicillin alone, what will happen? It will be secreted, and then from the tubules it will reach the urine, and it will be eliminated from the body. Okay, because of which penicillin has got a very short half-life. But if we combine it with probenecid, what's it going to do? It's going to inhibit the secretion. So the amount of penicillin that is excreted in the urine dramatically reduces. As a result, blood concentration of penicillin will increase. So it's a very clinically used drug interaction, okay, penicillin with probenecid. So what does probenecid, what is probenecid doing? It is involved in the secretion of drugs through the organic ion transporters, okay. So organic anion transporters actually secrete probenecid for elimination. But this probenecid itself has got this interesting clinical application. Okay, so carrier mediated transporters can there are these carriers, and there is another carrier that we need to remember, and that carrier is ABC transporters. Okay. Okay, so this is number 1 and this is number 2. Okay, and what is this number 1 and 2? They are involved in carrier mediated transport. Okay, so what is ABC do? So what is ABC actually? Let's learn here. So ABC is an acronym for ATP binding cassette transporters. Okay. Okay. So we now know what is ABC. ABC means ATP binding cassette transporters. Okay. So these ABC transporter, the most important example of ABC transporter which is important clinically is PGP. What is PGP? P-glycoprotein. Okay, so the P means refers to permeability. Okay, so it's permeability glycoprotein. So what does this do? The PGP is an efflux pump. Okay, so something, if suppose this is a cell and if there is an PGP here, so what the PGP does is it removes certain things from within this cell to outside this cell. Okay, so that is efflux. The opposite, if something brings in, we call it influx. Okay, but PGP is an efflux pump. It pumps out. It is especially important for anti-cancer drugs. Okay, so suppose a patient is given anti-cancer drugs like etoposide or doxorubicin or vinblastine or Geftinib. Okay. So if a patient is given this, any of these drugs, it gets into the cancerous cell, and this cancerous cell has got this P-glycoprotein, and that P-glycoprotein will remove all these drugs which have entered into this cell. Okay. So to kill this cell, you need these drugs inside the cell, but what happens is, whatever drug enters into the cancer cell is removed by the PGP. As a result, the cancer cell will demonstrate resistance. Okay, so so to remember, P-glycoprotein is involved in resistance. Okay, mediates resistance to anti-cancer drugs like etoposide, doxorubicin, vinblastine and Geftinib. Okay, but to remember that in carrier mediated transport, the most important transporters are solute carriers And solute carriers come in two flavors, and ABC transporters, the most important ABC transporter being P glycoprotein. Okay, so with this, we have in pharmacokinetics, we have with this, we have completed our discussion on membrane transport. Okay, so that is the first topic that we need to cover under pharmacokinetics. So we have covered membrane transport. Okay. Yeah. Let us now look at absorption. So absorption is defined as the movement of a drug from its site of administration into the central compartment. Okay. So it's the movement of a drug from its site of administration into the central compartment. Okay, so let's put this in perspective. Movement of a drug. Okay, so drug is usually not administered in its native state. Okay, for example, if we say aspirin, now aspirin is a drug. So this is a very bitter powder. Okay, so it is not administered in this form to a patient. Okay, because the patient might not find it palatable. Okay, instead, what's done is this active principle, this bitter powder, is converted into a tablet. Okay, and this tablet is then administered to a patient. So this tablet now contains, in addition to aspirin, it contains inactive constituents, means pharmacologically inactive constituents, which are called excipients. So excipients with this aspirin would be called API. What is API? Active pharmaceutical ingredient. Okay, so the drug is combined with excipients and made into a tablet. Okay, so the process by which the active pharmaceutical ingredient is mixed with the excipients and made into a tablet, that is called formulation. Let me repeat that again. Formulation is the process, all the processes by which the active pharmaceutical ingredient is mixed with excipients to form the tablet. So this tablet is called a dosage form. Okay, so this tablet is called a dosage form. So now we can say what is formulation. Formulation is all the processes which are involved in combining the active pharmaceutical ingredient with the excipients to form a dosage form. Okay, so drugs are administered to patients as dosage forms. Okay, so this tablet, so let's say this tablet is administered to a patient. Okay, so what is this tablet? It is a solid dosage form. Okay, it is administered to a patient because it's a solid dosage form in the form of a tablet. It is administered by a route of administration. It is administered by a route of administration to the patient. What is the route of administration here? Here the route of administration is oral. Okay, so this tablet is administered by the oral route to the patient. Okay, so after the tablet is ingested, what happens? The tablet reaches the stomach where it mixes with the gastric fluid, and then this tablet undergoes physical changes. What is the physical changes? It undergoes disintegration. Disintegration meaning it breaks down into smaller particles, and these smaller particles dissolve. Okay? Only after it gets dissolved, it gets absorbed. Okay, so what is absorption? So let's, we can draw it somewhere here. So we have got this dosage form that is administered to the patient. The patient ingests and it reaches the stomach and it undergoes disintegration and dissolution. Okay, so first it undergoes disintegration, then it undergoes dissolution, then it gets absorbed. Okay, what do we mean by absorbed? It crosses the intestinal membrane and gets into the blood. So it is absorbed and it reaches the central compartment. Okay, so the free drug reaches the central compartment, and what is the central compartment here? It's blood. Okay. This dosage form contains what? It contains the API plus excipients. Okay, the active pharmacological ingredient and inactive ingredients which are necessary to make it into a dosage form. So in disintegration and dissolution, what is happening is the active drug or the API is liberated from the dosage form. So therefore this step is called liberation. Okay, so what is liberation? It is the release of the API from the dosage form. Okay, so the drug gets liberated. After it gets liberated, it gets absorbed. Okay, so after liberation, it gets absorbed. So what is absorption? Absorption is the movement of a drug from its site of administration into this central compartment. And what is the central compartment? It is blood. Okay, so this process is called absorption. Okay, okay, so let's repeat it again. A patient consumes a dosage form. The dosage form goes into the patient. It undergoes disintegration and dissolution, assuming that the dosage form is a tablet. Okay, it undergoes disintegration and dissolution. By that process, what happens? The API is released from the dosage form, and this process we call it liberation. Okay, so after liberation, what happens? It gets absorbed. So any drug, it has to first, if it is in the form of a tablet, has to undergo disintegration, and then it has to dissolve in the fluid, dissolution. Only after dissolution it gets absorbed. And the what is absorption? It is the movement of the drug from the site of administration, the oral route, into the central compartment. So the free drug, after dissolution, it gets into the central compartment, and that is called absorption. Okay, so now we are going to study what are the factors influencing absorption when we give it by the oral route. Okay, so first we are going to concentrate on the factors that influence absorption when it is administered by the oral route.